Good evening, everyone, and welcome to the Brian Crombie Radio Hour on Saga 960. I've got a really interesting gentleman and fascinating technology and a launching company to introduce you to tonight. Howison Schroeder is my guest tonight. He's one of the most interesting, innovative entrepreneur, finance guys, scientist guys that I've ever met. He's one of these guys that's got a really interesting finance background that came across some science that he wanted to commercialize, and he ended up having to learn a whole bunch of science, and one of those situations where he learned so much that I actually wonder if he is a scientist, but he's not. He's just a finance guy that found the interesting company. I came across him in one of my prior lives, and he's still knocking on the door of success with this incredible opportunity to take a neurological drug now, a pharmaceutical product, to solve some concussion and other neurological problems. So, Howison Schroeder, welcome to the show, sir.
Well, thank you, Brian. Nice to see you again.
Nice to see you, sir. And I love your walls filled with gorgeous prints. So, tell me, what's the name of the company that you've launched, and what's the secret sauce, the technology of the pharmaceutical product?
The company's called Neuro Innovators. It's an LLC. The secret sauce really is the team that we've got. We've got a phenomenal team of people that have been through many of the wars. We've got a neuropharmacologist who was a professor actually at the University of Pittsburgh here, where I am, although I'd never met him when he was here. Pharmacology. He ran neurodrug development for Merck for a while, ran neurodrug development for Teva for a while. He's now over living in southern France, so we're anxious to have a company meeting over there someday soon. Another gentleman by the name of Mark Cochran, who's a Canadian. He actually was a partner in an early neuroscience investing VC fund called Neuro Ventures. He ran the Rockefeller Neuroscience Institute down here for a little bit. Was at Hopkins helping to commercialize technologies out of their medical school. Great, wonderful guy. Both are great, wonderful people. Got another doctor from Tufts who's a primary care doc who's been creative and inventive with me in the past, and actually another colleague who she worked for a pharma company, worked with me at Neurokinetics for a while, who good on clinical trials, or Coleman. Great team. Anyway, what we've done is in the neuro space, it's a very, very tricky place, which you probably know, to launch a drug. The success rate for new molecules pretty lousy. So we've focused on a couple of things. One, you mentioned concussions, but actually concussion still is a highly subjective metric to evaluate whether or not you have a concussion, particularly one that's got lingering symptoms. So the indication we've chosen is to help people with chronic motor deficits due to a stroke. Because if I can get you to move your arm 10% more, I can measure that and measure that objectively. So that is one of the key de-riskers here, is to lower what that outcome metric is. The other is using existing drugs, which you probably know more than I do about, and following a 505(b)(2) process. But with neural drugs, even when people have a modestly successful drug, single molecules have a hard time being clinically effective. They may be therapeutic, but you got to get so much more of it into the brain. So we've done a multi-drug prong strategy. So we're affecting multiple mechanisms of plasticity at the same time. And it also sort of lowers the amount of any one drug one needs to get in there. And we've just signed a research agreement with Harvard's Spaulding Rehab Hospital to get our concept into patients as soon as we can finish our seed round here.
Excellent. And so how many different pharmaceutical compounds are in this cocktail?
There are 3 in this cocktail. We've identified and filed a patent on a little over 152 different drugs, which are well supported in the literature to promote some form of neuroplasticity. One example would be fluoxetine, Prozac, bupropion, which is one I think you know well. But it's interesting. People have tried to use single drugs like this for neuroplasticity, but it hasn't really worked yet. And our theory is that it's just because it's just too limited a mechanism of action in a widely varied human population, and B, in a very, very, very complex structure like our human brain.
So you're at early stages. You're going to do some testing with this hospital at Harvard. If it works, what's sort of the progression? What's the time period? How long would this take before people actually get to see it on the pharmaceutical shelves?
We think it'll be pretty quick. We think we could have it on the shelves within 5, pretty safely, we think 6 years. So the rehab that's been done for people that have motor deficits from stroke, and actually we're looking at the people that are 6 months or more past their stroke. Interesting little bit of sort of biologic trivia. When we have a stroke, our blood-brain barrier opens up. That's the barrier that filters stuff in and out of the brain. And so the big molecules, the growth factors, have a better chance of getting in and helping the brain actually heal, repair itself. Then it shuts down. And we're targeting the period after the blood-brain barrier has gone back to its sort of normal state. We've seen in a number of different studies that physical occupational therapy actually works well if it's intensive and people stay with it. So what we're doing then is basically trying to put a little bit of, it may sound a little crude, but a little bit of miracle grow for the brain in there to expand and accelerate that growth. So when people do that physical rehab, 2 months roughly. So we're going to know within 6 months whether we've got a grand slam or maybe just a single or two.
Really, this is interesting. So it's not something that you would treat within the first 6 months after a stroke, but after the 6 months you would, and it's combined with intensive physical therapy.
Correct.
And what's, just out of interest, what's the, do you have a double-blind study? What's the other arm? Is it people that aren't doing intensive physical therapy or just not taking your cocktail?
So the double-blind part of it is just basically the historic controls that people have already done with these studies. So Dr. Bonato at Harvard has run many of these studies. He's also actually dabbled in some of the single neuroplastic drug ideas to see if that would have an effect, which has made him a hugely beneficial partner in this study. So the consensus is, you know what, there's so much data about how much people can recover without a drug that if we put the drug in, we feel like we're going to get a pretty good signal as to where we're going to go, then move to a dosing study to make sure we kind of refine it and come up with the perfect sort of single pill package.
This sounds fascinating, Howison. If people want to check out your company, is there a website they can go to?
Yep. Neuroinnovators.com or niv-neuro.com. And you can see the link on my LinkedIn page too, if you wish.
Neuroinnovators.com. Fascinating. We're going to take a break for some messages and be back in just 2 minutes with Howison Schroeder talking about neuroplasticity, which is an interesting topic, and a drug cocktail that he's working on that's going to help people after they've suffered a stroke regain motor skills. Stay with us, everyone. Back in 2 minutes. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. I'm chatting tonight with Howison Schroeder. He's coming into us from Pittsburgh, Pennsylvania, United States. He runs a company that is it's not called Neuro Innovators, but your website is Neuro Innovators. What's the company called, sir?
No, it is called Neuro Innovators. That's
Neuro
right.
Innovators. Okay, I apologize. And you're developing a cocktail of pharmaceutical products that are older pharmaceutical products, because you're actually taking them through the approval process through something with the FDA called a 505(b)(2), which allows you to use drugs that have been previously developed by someone else have now become generic, but you're going to prove that it works in some other area, which is, I think, fascinating, and it is a route of approval that I'm familiar with because it's something that prior company I worked with followed 505(b)(2) strategy all the time, and you're trying to solve, a conundrum that we have, which is that people that are suffering from a stroke or have had a stroke lose their motor skills and motor activity. There's an interesting company that I've interviewed in Canada that uses electrical stimulation to try to get people to regain the ability to control their muscles, their joints, movement, etc. So maybe we should talk about at some point in time some of the other ways that people are trying to solve this problem. Maybe we could take a step back though and talk about how you came to where you are. You and I came across each other when you were running a company that I think at the time was called Neuro, and you were developing a machine, a medical device at the time, and one that actually in one version was a chair that people sat in. Tell me a little bit about that part of your business career, if you could.
So that company was Neuro, you're right. Actually, when I got there, it was an interesting, almost just sort of a engineering consulting firm for motor stimuluses for people. So the basic gist is our vestibular organ that's in our inner ear is how we sort of manage and control and are aware of ourselves in space. So as we walk around, it tells us how we're moving forward, bouncing up and down, whatever we do. So the point there back in the early NASA days was, if I'm going to spin you around or put you up into space, how are you going to function? And so a test was developed called a sinusoidal harmonic rotation test, which means I'm going to spin you around, but I'm going to measure and monitor what your eyes do in response, which is called the vestibular ocular reflex. Well, we sort of dug that company out of bankruptcy, got it going, and we did that by focusing on the eye data. And there have been multiple ways to measure the eye, electrodes, et cetera. But we focused and drilled down and dug deep into the digital video technology that was really just, beginning to explode when we got there. And we built off that. Some people used to call it the big blank chair. We built off of that because there are a plethora of other little tests that you use. Smooth pursuit test, where the doctor moves their finger back and forth in front of your eyes. And if you did this battery of tests and you measured the eye with exceptional sensitivity, you're basically measuring the neurofunctional performance health of the brain.
But hold, this is interesting. So what you're saying is that by tracking the eye and the eye's movements, you can actually test not just the eye, but the neurofunctionability of the brain.
Correct. If I do, for example, if I do a horizontal smooth pursuit, is the technical name.
Which is a doctor just running his finger or her finger back and forth in front of your face.
Correct. And then
watching your eye movement.
Right. And then if I do a vertical one, I'm using different parts of my brain. So if I'm normal here, but abnormal here, that gives me some sense of where in the brain I've got the problem.
And then what you've done is you developed something where people would put glasses on and actually watch a ball, a light, and you would use a camera to track their pupils and how their pupils reacted to that moving ball, correct?
And the pupil, so then there are two things about the pupil. One is just the pupil changing, which is actually hugely informative, as well as kind of a challenge when you're trying to track the eye. And the other is, where is the pupil looking as the eye is moving around in your socket? And you, me, we humans, we process images at about 30, 40, 50, maybe 60 frames per second. So most of the screens we're all looking at, many of them refresh at 60. It seems, benign to us. I think our TV's at about 30. But your brain can move the eye at something much closer to, depending upon what sort of activity you're looking at, 250, 500, 1,000 frames per second. So you've got to have technology to actually be able to measure what the brain is really doing. And if you are at that level of sensitivity, far better than the average human, you can actually see these diseases, like multiple sclerosis in particular was one that got pretty well mapped. And we could see it before the MRI could. The
Through
challenge
the
was
movement of the pupil.
the movement of the eye, basically in the pupil, and the eye's response. So a certain profile of abnormal responses to this battery of tests could be a very strong indication that one has MS long before you can see the lesions in an MRI. But now correlating that data to the MS and to the MRI, et cetera, it's you got to run a lot of big ass expensive clinical studies.
Well, but it was interesting when I saw your technology, because what I think you showed is that there was like a bell curve of humans' reaction time, and it was interesting because what I think it showed is that successful pro athletes, had a far quicker and better eye movement, and they were on like the 95% portion of that bell curve of a human's ability to respond. And it was interesting because one of the smart scientists that worked with you at one point in time told me that some of these very successful athletes just see things faster than the rest of us.
Correct. So there's a simple thing called a saccade test. A saccade is a very fast jump of the eye to a target. So one of the tests we would do is present a dot somewhere on this screen, and then we watch the eye jump to that dot. Most of us have what we call a primary saccade or primary eye movement, and then a little catch-up or secondary eye movement. When you test some of the people in special forces or test some of the athletes, they don't have that little secondary movement. They go right to the target. So an interesting example that people seem to grasp, if I'm a special forces guy and out there on the battlefield, I get my target and pull my trigger quicker than the other guy. Now, the other guy might get there and pull the trigger and we might both die, but the other guy's gone because I can put my eye on that target and pull the trigger faster with sufficient accuracy. So yeah, we're all different and makes a difference. Now, I can make you better. I probably can't take you from 0 to 100, but I can probably take you from 70 to 90.
Through training.
Through training.
Really, that's interesting. I was watching the Super Bowl last week, and it was interesting because the announcers kept on talking about the quarterback's spatial awareness. And I was wondering whether you could actually test that, because I was thinking they just see where the runner is and is going faster than other people.
I'm sure there could be a way to test it. I don't know how one might do that, but yes, I mean, some people have an ability to have a very wide visual field, so to speak, cognitive visual field.
Unbelievable. And then that company ended up being sold, but you're still involved in the area. So I think that's interesting. Let me ask you a couple of other questions just for background, so people understand. You said neuroplasticity a couple of times. What, sir, is neuroplasticity?
So neuroplasticity is the ability, capacity, process of the brain to rewire itself, repair itself, restore its function. So let's take a stroke. If I have a stroke, an ischemic stroke, basically I've got a little spot in my brain that lost blood for a while and effectively died. So there's some neuroinflammation that will go in there. There's a little corona around that, but I've lost that little part of my brain, access to it, use of it. If we can actually restore that, either rebuild new connections through that little spot or help the brain wire more aggressively where it needs to compensate for that action. So the brain, there's nothing we do with our brain that is done in any one area of our brain.
Sorry, that's interesting. Say that again, please.
There's nothing that we do when we're using our brain that is done in one single area of the brain.
Really, that's interesting. So when we move or think or talk or do whatever, it's several parts of the brain that are involved?
Correct. Now there's a primary area, but then there are 2, 3, 4, 5, 6 different backup areas. Actually, there was a wonderful researcher at McGill years ago who, Kathy Cullen, who actually sort of really, pun intended, I guess, hit a nerve. She was operating on some creatures and she cut a specific nerve where she expected that animal to lose function for a period of time before all those backup systems would kick in and restore that utility. When she cut the nerve, it never lost it, didn't lose a nanosecond of function. So these systems are working all the time. So if I lose some part of my brain, there are backup systems that are operating pretty quickly. If I can now help them sort of restore or compensate, then I can get some of that function back quicker.
That's interesting. So what you're talking about is not actually repairing that portion of the brain where the cells died, but you're activating the backup system portions of the brain.
And this is where we, unfortunately, there's something amazing about humans. We don't like folks cutting into our brains to inspect that. It's okay for our livers or our lungs or other organs in our body, but not our brain. We expect that we will be doing both. And this is where some of the engineering of our drug compound will come into play. The drug compound we've chosen right now is really, it's designed to get ostensibly a rapid, low-risk clearance from the FDA. We think there's some other drugs out there that could actually begin to build on this concept and this model, but they're going to take more time and more money and have slightly greater risk with the FDA, which means more time and money on the clinical side. So we've really engineered this to be as really as low risk a process through the FDA into patients as we can. And as we begin to study patients, what we're going to be able to learn is just going to be fascinating.
This is fascinating. It's really quite interesting. And, what you described previously to me is that there's numerous different things that have impacted the brain that your prior company could assess. And it wasn't just multiple sclerosis, but it was concussion, it was Alzheimer's, it was dementia, a whole raft of different issues. Do you think your drug is going to be active in more than just stroke?
So let me back up a little bit in two things. One, the quick answer is yes. And one of the reasons why we got into this is because I didn't think we were capitalizing on this objective data set we were getting from the eyes. And I really did not think that the pharma companies were capitalizing on that. So as I went through this process, again, the objective metric we get from chronic stroke, we don't need something that the FDA isn't familiar with. They understand this process. So as we anchor knowledge, yes, when you deal with neuroinflammation, you're looking at stuff like Alzheimer's. We've got some ideas for that. We've got some ideas for TBI, but we've got ideas for MS. All these neurodegenerative diseases, particularly if they're injuries like a stroke or TBI, but also the ones that are progressive. Yes, but you got to walk before you run. So let's get the first disease anchored and get the concept sort of rooted.
But before you go on, let's just make sure everyone understands some of those terms. TBI is what, sir?
Traumatic brain injury.
And MS?
Multiple sclerosis.
We're going to take a break for some messages and come back in just 2 minutes. And we're going to delve into and try to get a little bit more in depth with Howison about what this drug cocktail is and why it might actually be having this positive impact. Stay with us, everyone. This is a really interesting technology company, and I got to tell you, individual. This is one of these guys that amazes me that he starts out in finance and investments and falls in love with a therapeutic area and issue, and delves into it and becomes so knowledgeable that he almost is equal to a scientist. It's just
okay. Amazing. My head's getting big now, Brian. Stop.
Justifiably, because you are one of those guys, and you're able to take some of the complex stuff that the scientific people say and dumb it down so a guy like me can understand it, which is a real skill. Stay with us, everyone. We'll be back in just 2 minutes with Howison Schroeder. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. It's really interesting for me tonight to be chatting again with Howison Schroeder. He runs a company called Neuro Innovators out of Pittsburgh, Pennsylvania, that is developing a cocktail pharmaceutical product that he thinks will assist people in recovering from a stroke and regaining their motor abilities. And it's interesting. And he's in the first stages of the clinical trials right now with one of the hospitals associated with Harvard University with this cocktail. And he's got an interesting approach, which is a 505(b)(2) strategy, which allows companies to use previously approved drugs, pharmaceutical products that have now become generic, and therefore rely on their previous safety and other efficacy data to not have to go through everything all over again, but prove they work in a new area. And I think this is a fascinating strategy, a smart strategy, a quicker strategy, and I hope it works, because it'll be fantastic if it does. Howison, let me ask you a few questions that have got to come to mind. You mentioned that you were using fluoxetine, which is known by the brand name Prozac, and bupropion, which is known by the name Wellbutrin. Both of those are antidepressants, and they work on different mechanisms of action, but they're antidepressants. Why would an antidepressant help the brain repair itself or reconnect with different backup systems in the brain?
Well, one, I've thrown those two drug names out as just sort of examples of drugs on our list that have been shown to promote some mechanism of plasticity in the brain. And the I can't give you the good, solid neuropharmacological explanation of why the SSRIs, which are the family of antidepressants, work or don't work well. I don't know that we really know a lot of that. We know the mechanisms of action, but so we've actually picked 3 other drugs, which I gotta keep confidential for a bit. It'll become public once we get this clinical study up and running. We're gonna have to disclose it to the patients at a minimum. But it is fascinating, the different kinds of drugs that have some demonstrated neuroplastic property, vasodilators, diabetic drugs, and we can speculate as to why they seem to work, which is actually a conversation we're having somewhat regularly amongst ourselves to see how to make sure we engineer our future for the company smartly. But the 3 that we have now are all 3 in different sort of families of drugs in order to make sure the variety, the sort of multi-vector strategy of attacking multiple different mechanisms of action. And we've maybe getting a bit too in the weeds, but the mechanisms of action we're talking about are apoptosis, which is cell death in the brain. Neuroinflammation, that's an obvious one, right? We don't want to keep the brain inflamed. We want to calm that down. But take neuroinflammation. One drug may do a better job with interleukin-6. Another one may do a better job with interleukin-4. Another one might do a better job with C-reactive protein. And by
I got to interrupt. What is interleukin-4 and interleukin-6?
Yeah, well, now I'm really stepping outside my my bounds where I need to go back to hide under my finance hat. But these are different inflammatory proteins in the brain and markers. So if your interleukin-6 is going up, you've got greater neuroinflammation.
Fascinating.
So I can't get more detailed than that.
That's fine. So these different pharmaceutical products have different mechanisms of action, and you think the cocktail altogether will assist in the neuroplasticity and the repair of the brain, and the inflammation, bringing down the inflammation of the brain, which sounds fascinating. Let me ask you a few conceptual questions, and if you don't know the answers, that's fine, but it's interesting. So, fluoxetine, Prozac, was also indicated for smoking cessation. And the hypothesis there was that it took away some of the sort of addictive natures of the brain. And Wellbutrin, bupropion, was for weight loss, and so it gave people, the impression was that it gave people more satisfaction or happiness, or less strife with their life, and so therefore they didn't eat to feel good. Why would something that makes you want to stop smoking and or stop eating help repair the brain?
I'm going to answer it maybe a little obtusely, Brian. So let's go back to neuroplasticity for a minute. So the term actually most recently has been a bit usurped by the hallucinogenics. Those people who are working on psilocybin, LSD, lysergic acid, ketamine, etc. And a really good friend of mine who used to run the neurotrauma portfolio at the Army, he's now consulting for him on their drugs. He basically described the psychedelics as gate opening drugs. So we've got idle pathways in our brain, and we've got active ones. And we through habituation, whether I get addicted to smoking or addicted to eating, there's a pathway that we get into a rut and we stick to it. With the psychedelics, when they open up all the gates, and we've seen Johns Hopkins in particular, I think, has some success in this, but there are a number of institutions that are using psilocybin now with concurrent psycho sort of guidance to get them off of their addiction from alcohol or other drugs. And it seems to be working quite well. So open the gates, use those other pathways that have gone idle, and change behaviors. And I don't know Wellbutrin or Prozac well enough to give that specific answer. But what we're doing is actually working to sort of repair the brain, but without any of the hallucinations. We're not really necessarily opening up pathways. We're working to sort of activate or calm down the challenges in the brain as they are. I'll add one more thing. I think when you and I were growing up, every time we went out and had a beer in college, somebody told us we were killing brain cells that we would never repair.
Yep.
That thinking has now gone away. We don't.
Good. That's helpful.
Yes, it is. We don't lose neuroplasticity. It just drops dramatically as we age, but we can still create new neurons, not often, but we can do that. We can absolutely build new pathways and new oligodendrocytes, which are different mechanisms of communication, sort of structural communications in the brain, et cetera. Anyway, I'm not sure I quite answered your question, but I'm not sure I can really answer.
Well, I got to tell you a story that's relative to this. I sat down with an old boss of mine just a couple months ago, Jimmy Pattison, who's a $9 billion billionaire in Vancouver, British Columbia, in his 90s, mid-90s. And I asked him what the secret was. And he says, Brian, never stop doing deals. Because as soon as you stop doing deals, you're going to stop using your brain, and then life will be over. So keep doing deals. So Howison, keep doing deals, buddy. Keep doing deals. Let me ask you a few other questions if I could, because you and I have had some of these conversations previously. I worked, as you may remember, once on IVIG, which is a portion of blood plasma, effectively, and we were with a company that was taking a look at, I think it was called, artificial ways of stimulating some of the blood plasmas, and they found that the actual blood plasmas, even if separated, worked far better than any of the artificially created ones. And the hypothesis was that you can never get down to just one thing. The cocktail of the blood plasma was always more successful, because there were other things that were involved in it that helped. And if you only had one, you were only having impact on one mechanism of action. And then certainly with all the AIDS and HIV drugs, again, cocktails ended up being the solution. So is there some secret in this, your use of a cocktail and my example of an HIV drug's a cocktail, and in blood plasma a cocktail? Maybe the body's complicated, the brain's complicated, and there never is just sort of one silver bullet that it takes a cocktail.
I think that's just spot on. And the FDA, our system, our sort of biologic science system is, and you as a finance economist kind of person will appreciate this, but it really is, it's in an effort to de-risk, we've drilled it down to such a single variable science. In economics, we use the term ceteris paribus, all other things being equal. And it effectively allows us to look at one or two, three mechanisms and see how they interact, and then move to the next two or three. We're going to hold those equal and see what happens. I don't think our regulatory system and much of our biologic science has that same sort of flexibility and dynamism that the dismal science of economics has. But that is so spot on, and that's exactly what we're doing. And the FDA is slowly moving towards these sort of accepting these compound drugs. The AIDS one, that was the first one. They're doing it more and more with cancer now, as you well know. Nobody's really tried this in the brain yet, so there's a risk that we're taking. And it's one of the reasons why our first study is basically an investigator-initiated study. We'll be looking at these drugs pretty much off the shelf. It's a very complex dosing strategy. But then when we move into the FDA, we'll have some data, and frankly, the population of people is huge. So with a huge unmet need and successful improvement in people's motor function, and what we expect to be very little, if any, side effects from the drugs, off we go with the FDA and hopefully get through that compound challenge.
Now, you're a private company, you're not public.
Correct.
And so if people are intrigued and want to invest, is there they have to probably be of a certain net worth before they're allowed to invest? What's the regulatory process one might have to follow to invest in your company, sir?
So we've got two accelerators here in Pittsburgh that have committed $400K. That has to be matched in one way, shape, or form. We have another group out of Charlottesville, Virginia, called Clockwork. Clockwork is basically putting together a special purpose vehicle for smallerish investors. Otherwise, just come straight to us. Go to our website. There is an email there. You can go to my LinkedIn page and also kind of reach out and connect to me, and I'll make sure you get in the right place with the right sort of support and documentation. You might also find my white papers there on neuro investing, which drills deeply into this sort of objective metric issue with the neuro drugs. But my email is h.schroeder@neuro.com.
Neuro Innovators is the website.
Neuro Innovators is the website.
We're going to take a final break, and come back with Howison Schroeder in just 2 minutes and some concluding comments. Stay with us, everyone. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. I'm having a really interesting conversation tonight with Howison Schroeder, who is the founder and CEO of a company by the name of Neuro Innovators that is developing a pharmaceutical product that's really a cocktail of several pharmaceutical products that he thinks when taken will help stroke victims, people that have suffered a stroke 6 months after they've suffered a stroke, and along with intensive physical therapy, have them regain motor skills and motor ability and be able to control their body again, which I think would be spectacular, because I think all of us know some stroke victims that still have active brains and still want to live an active, normal, regular life, but can't control some portion of their body anymore. And it's got to be like unbelievable to not be able to control a part of your body that you used to be able to control. I can't imagine what it would be like. And so good luck, sir, and Godspeed in that regard. I hope your clinical tests work, and I hope your drug gets approved, and I hope it has the impact that you think it's going to have, because it's going to improve people's lives. But we first met when we were talking about concussion, and it was interesting. And one of the first things that you told me to do was to watch a movie by Will Smith on concussion to realize how challenging that was. And I was really affected by, how traumatic concussions can be, because we think about them as something everyone suffers. And you identified TBI, traumatic brain injury, which is, I guess, bad concussions. And I've met and know people that have fallen off roofs and ended up, losing their mind effectively, having the mind of an 8-year-old again, and or people in car crashes, or, sports athletes that, end up being negatively impacted by repeated concussions. What do you think, after what is it, a decade, more than a decade in this area? What do you think the opportunity is to find some solution to either diagnosing a concussion and or repairing a concussion?
It's diagnosing concussion, sort of a big topic when we're just really have to learn how to measure the brain. Objectivity becomes key. And sort of back to this conversation about multiple mechanisms of action, people trying to get the saliva markers and various other biochemical traces of a concussion. I think that's a tricky game. Obviously, Abbott's got a commercial product that they've released, and I think having some success with. You can't really get into an MRI, too expensive, doesn't get around. So what's portable? And when we look at concussions, any medical device, concussion just being an indication with an example, you've got to be looking at the point of care and the cost and sort of utility of that device in that point of care. And it gets so complicated when you do that. To identify a concussion on the side of a ball field versus identifying a serious traumatic brain injury in an emergency room, you don't need a device in the emergency room. Some of these friends you mentioned, they fall off their roofs, and it's pretty obvious what's happened. Having your high school son, daughter in an ice hockey or football game get their bell rung. Well, gee, is this life-threatening, or is this just part of the natural injury process we humans go through, and we heal from most of it and keep moving? EEG has been the long standard for neurologists to evaluate brain function. Boy, that's noisy data. It's very difficult to get signal from noise. And I'm sure at some point AI may well help us there. But, how do you come up with something objective? I think eye tracking is still good eye tracking, and very few do good eye tracking. Good eye tracking provides the sensitivity that allows us to get to the specificity, I think, today. There's a clever little test that I think can work. It's a company called Cognitivity out of the UK. They've got a wonderful little test, but they've designed it for dementia, not concussions. I think they should be moving into concussions, because I think it's a great idea to help triage that. But, that's anyway, that's a rambling answer, Brian.
What's the name of that? What's the name of that company? I want to follow it up.
Cognitivity.
Great name. Howison, this has been really interesting. Thanks for reconnecting. I really appreciate hearing what you're up to. Good luck.
Thanks, Brian. Appreciate it. Nice to see you again, bud.
Nice to see you. Remind everyone, what's the website if they want to go to it to check you out?
Neuro-Innovators.com.
Neuroinnovators.com. Howison Schroeder, good luck. I hope you solve the problem of people's motor function after stroke. And if anyone can, I think you can. So good luck.
Thanks, Brian.
That's our show for tonight, everyone. Thank you for joining us. I remind you, I'm on every Monday through Friday at 6 o'clock on 960 AM. You can stream me online, even from Pittsburgh, Pennsylvania at www.saga960am.ca for Canada. Stay with us, everyone. Back next Monday through Friday at 6 o'clock.